Bioinfocongress IV Associate Professor Dr. Ferhat Ay Computational Methods for Studying the 3D Organization of the Human Genome. Associate Professor Dr. Ferhat Ay received his bachelor's degrees in computer engineering and mathematics, both from Middle East Technical University, Turkey in 2007, and his PhD in computer science at the University of Florida in 2011. He subsequently joined the Department of Genome Sciences at the University of Washington as a Computing Innovation Fellow, awarded by Computing Research Association and National Science Foundation. He was a research assistant professor in the Department of Preventive Medicine Health and Biomedical Informatics at Northwestern University in 2015. Before establishing his lab at the La Jolla Institute for Immunology in 2016. He is currently the Institute Leadership Associate Professor of Computational Biology at LGI, an adjunct faculty at the UC San Diego School of Medicine and UCSD Bioinformatics PhD program. His primary research areas are bioinformatics, computational biology, epigenomics, and regulatory genomics. His lab works on understanding the relationship between three-dimensional genome architecture and gene regulation in the context of different immune-mediated diseases. He serves as an associate editor for PLOS Computational Biology and an editorial board member for Genome Biology and the Journal of Computational Biology.
Dear Associate Professor Dr. Ferhat Ay and participants, welcome all of you to our Bioinfocongress IV event. Associate Professor Dr. Ferhat Ay will give us a talk today on computational methods for studying the 3D organization of the human genome. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Ferhat Ay, welcome back.
Thank you very much. Thanks a lot, Nazlı, and thank you to the whole Bioinfo Range team. What you guys are accomplishing is amazing. I think you guys are showing a good example as what you can do with little bit, only little resources, but maybe the youth energy and the willingness that you guys are showing for organizing such events and teaching a lot more people in our country bioinformatics and related biomedical sciences. I should probably start with my slides, but you think that the aspect ratio, if this is small, then I can just probably turn this one off. I'm sorry, I was trying to do it with 3 screens, but maybe this is just too much. So I'll just share another screen. I just wanted to see you guys and the questions all at the same time, but this might be a little bit too much. Hold on 1 second. All right, how about now? Can you guys see the slides? Because I cannot see you right now. Can you guys see them?
Yes, hocam. We can see
okay,
clearly.
Perfect. All right. Yeah, sorry that I tried too much, I think. Thanks again. Thank you all. And thank you for staying. I know it's late night almost in Turkey. And yeah, but I'm on the West Coast, so it's kind of like in the morning for me. I hope that you guys are still able to stay awake and listen to this talk. So as the introduction mentioned, I'm a faculty right now at the La Jolla Institute for Immunology and also at the UC San Diego. And what we study is the 3D organization of eukaryotic genomes in general, but mainly human. So before I really go into any of the science, as you all know, I don't need to, and I think, you know, there's nothing that hasn't been said about what's been happening in Turkey. So I'm not going to belabor, but we've been having one disaster after another. And I know that everybody is still trying to hold on to what they are working on, their ambitions, their hopes. So I really hope that everything is going to be much better for our country. But in the meantime, my sincere, condolences for everyone who's lost any family, any friends, including you guys. You've lost a team member. So I'm just hoping there's going to be better days ahead. So I wanted to start with a little bit of my journey, and I sometimes show this slide to people here too, just to tell them like what Turkey looks like and what is where, and also like in terms of you guys and in terms of young scientists, it's important that like I don't know, professors don't grow on trees, right? So we also come from real lives and real locations. So I was born in a small town in Manisa in Gördes. It's about 10,000 people. I lived there for a while. Then my family moved to Bursa, and then I joined the university in Ankara, Middle East Technical University. The first time that I flew abroad, it was for my PhD, starting my PhD, and that was University of Florida. So I was there for 4 years. I had a brief internship at MIT in the meantime, then moved for my postdoc to the other side of the country. This is about 3,000 miles away. And then after my postdoc and of course getting married there and starting a family, we moved to Chicago for a year and then to San Diego. So we have covered all the corners of the country. And then together with a lot of the big projects that we have in lab, we also have 2 small projects at home. So this has been my life in the past couple years, essentially, juggling science and kids and all the disasters and all the things that are happening around the world and in my country and in the USA. So what do we do in our lab? We are a computational biology lab. So we do have interest areas in a lot of different diseases and applications of computational biology to them. So some of them are related to cancer. With some pharmaceutical companies, we are working on autoimmune diseases, and also we are working on some infectious diseases. But what ties everything all together for us is the computational methods development. So developing methods that are of high utility and that are accurate and efficient for analyzing the ton of data that's being generated in labs all across the world, right? And then as you can see, the bioinformatics starts with bio, right? I'm a computer scientist. I did computer science and math and all of that. But now that we are doing bioinformatics, we always need to keep in mind that it starts with bio, and the biology aspect is really important. So with that in mind, we are happy with being a computational lab. But I mean, there are some disadvantages of computational work, and then I will only mention one here. This is a quote from my 6-year-old a month ago when I was going for a seminar. He's like, "Why are you going, Daddy? You're not even a real scientist. You just do stuff on your computer." Jokes aside, but that might be the sentiment sometimes computational biologists might be getting. But this is something that you should, never really worry about. Holding a pipette versus typing things on your keyboard, they are not inherently different. They are all about like discovering something that people have not discovered before, and making humans happier and healthier and more hopeful for the future. So what we do really like in the lab is to try to understand this intricate organization of how our DNA folds and fits into our nuclei in our eukaryotic cells, right? So we have 23 different chromosomes, 2 copies of each, and then they fold and fit into a teeny tiny nucleus, right? So when you add all the DNA on top of each other, kind of stitch them together, it's about 2 meters of DNA per cell, and then it's fitting into a nucleus that's about 1 million times smaller, right? So how does this happen has been a question that the field has been asking for many, many years. But the real revolutions and breakthroughs came when we went from being able to look at only under the microscopes to a few regions at a time to this whole next generation sequencing based assays where you can now look at the many cells all at once and then all loci, all regions, and then how proximal they are and so on and so forth. So the working horse in our field for the 3D genome and chromatin organization these days has been this assay called Hi-C, and there are so many variants of it. But essentially, it is you take your cell, you take cells, you take your sample, you cross-link them. So essentially, holding things together in the nucleus for a while. The things that are close to each other will be held together by these cross-links, and then you need to use some sort of scissors, whether it's restriction enzymes or some other versions of it, but you need to cut them so that you kind of like go through some sequencing steps after that. You cut them. When you cut them and don't remove the crosslinks, they are still together if they are in the 3D genome, if they are proximal. So after a couple steps, you can religate them, put some markers, pull down with those markers, etc. You need to linearize it so that you can sequence. But once you do that, and once you, of course, spend a lot of sequencing dollars essentially on these libraries, you may get hundreds of millions or billions these days of read pairs, where each read pair will tell you what is the proximity between, let's say, these yellow region and orange region in the genome, how many times you are seeing a read that puts them together in this one product. So our job starts after that. And then the nice thing about all of these, we can completely forget everything that I said and think about this as a matrix, just a mathematical object, right? And then when you look at this matrix at like different zoom ins and different resolutions, you can see a lot of patterns that explain how our nucleus is organized. So for instance, at the very broad resolution where you see all the chromosomes, of course, you see the chromosomes are tightly folded within each other, not as much interacting within the next chromosome and so on and so forth. So this we call chromosome territories. So you can take a chromosome, zoom in a little bit, and then you see these checkerboard patterns, flat pattern here. And then this tells you that some regions like to interact with others more than the rest. And then that there is what we call compartments, chromatin compartments, where some of these regions are active and they tend to interact with each other more, and then some of these regions are inactive in their chromatin. These are harsh generalizations. These are multi-megabase regions, of course. There's more fine-grained things happening there. But then again, if you take maybe one of them or two of them and then zoom in, then you start seeing these diagonal blocks. And sometimes you see these dots on those diagonal blocks. So this is what we call these blocks, is what we call domains or topological domains. And then these dots are what we call loops. So this one nicely depicts, the graphic underneath it nicely depicts that these loops can be between the two endpoints of such domains, or they can be within the domain and connecting regulatory regions such as genes, their promoters and enhancers, and so on and so forth. So in our lab, we have been very focused on the loop calling, the identification from these matrices, identification of patterns that are suggestive of loops, whether they are at the boundaries like this or whether they are connecting regulatory elements. So our lab has developed a series of tools in this for different data types, and as the resolution and the field has evolved. More recently, we have also gone back to this, the broader architecture, which is the chromatin compartments. Calling them has been not much of an issue, but comparative analysis is, and then now we have a ton of different data sets that you need really solid statistical methods to do this. We have just developed one recently. And then one other aspect of this we have worked on, and I'm going to talk to you about this also, is the genomic rearrangement. So you can think of them mainly in the context of cancer, when a linear DNA is kind of like shuffled around, when something is deleted, when something is amplified. What happens? And what happens is that some unexpected patterns are seen in these high-C maps, and we can opportunistically use them to discover such rearrangements. And this is very helpful in the context of cancer. So this is a classical example. I've seen this slide and I presented this slide, I mean, maybe hundreds of times right now, but it never gets old because it is amazing. So it is a gene called sonic hedgehog that is very important in development and in different stages in development. And it has different enhancers in different places. In one part of the development, in the limb development, in either mouse or human, there is an enhancer that controls this gene, and it is 1 million base pairs away. So that blows your mind when you think about what people are looking. When we generally look at what regulates the gene, you look at the gene and then a little bit around it, maybe 5-10 kb at most. This has been the status quo for many, many years, but now, of course, it's changing. There are very important genes such as sonic hedgehog that are being impacted by multi-megabase loops, such as, I'm just checking my email just in case everybody is here, but let me know if I disconnect. I'll have my email. What's happening here is that changes or deletion of this enhancer totally determines how your limb development is going to work out. And in humans, the same enhancer is conserved and also still quite far away. Even a single base pair change in that enhancer is changing the phenotype. You can see all these diseases where you have like a polydactyly and more digits than what is assumed normal. So what this is telling you is that maybe we have been overseeing certain regions such as these enhancers for many years because we haven't been looking at the right places. And the only way for someone who's rational to be even thinking about this enhancer as something to do with this is some sort of physical evidence that they are coming together in the three-dimensional structure, right? Otherwise, you cannot exert anything over such distance. So a little bit of background in the looping field and kind of where we are right now. So the loops and TADs, these domains, how are they formed? So in 2014, with the increasing resolution of these high-C data sets, Erez Lieberman's group was able to come up with a hypothesis that said, when we look at these strongest loops, what we see is the binding of CTCF, which is an insulatory protein, on the anchors of these strongest loops. They are highly enriched. And then what they showed was that not only they're enriched, but it's not palindromic, so it can bind to one strand or the other. So the orientation at which this CTCF binds it matters. And then only at the divergent orientation, the loops kind of like are the strongest here. So how is that happening? What is how do these two ends find each other? Right? And then came the idea of this loop extrusion, which is there must be something that's extruding the DNA, such as this slide that you're seeing here. And then maybe CTCF, these divergent CTCF sites are where this extrusion stops. And then you have this domain formed in the number 4 here, as you can see. And then they have tested this idea by deleting CTCF sites and adding CTCF sites, and then the predictions and the experiment, in at least a limited number of cases, have kind of really agreed, supporting the idea of this loop extrusion. So what is this loop extruder though, right? I mean, CTCF is where it stops, but what's this motor kind of thing that extrudes the DNA? So we think that it's cohesin. But before going there, people have looked at what happens when you degrade the CTCF. You cannot 100% degrade it, but if you can, get it to a minimal level, what happens is all of these loops and domains disappear, but nothing changes in terms of these compartments, these A and B compartments. Nothing changes in terms of replication timing. We have shown nothing changes. So when you go into cohesin, which is what extrudes these loops, and it's a huge complex, so you really need to look at its individual components and knock down some of its subunits. What you're seeing is again, it does eliminate loops. Compartments are not touched as much. In both cases, though, what has been puzzling to all of us in the field is that if this is so important, if all of these loops and domains are so important for gene regulation, then when you fiddle with the CTCF or QASN, then you expect a lot of changes in the transcription too. But that's not the case. The changes to transcription has been very minimal. And there are reasons for that I will not go into today, but happy to discuss with you guys if anybody's curious. Then people have gone into depleting what loads and unloads QASN. And then these have been very interesting, that if you deplete what releases QASN from the chromatin, then you have loops going on and on and on, stronger and longer loops. But compartmentalization is kind of like lost a little bit, that flat pattern is lost a little bit. Vice versa, if you deplete what loads QASN onto the chromatin, then you really cannot establish these longer range loops that you're seeing here. They are lost, but it kind of gets compartmentalized a little bit. So then came the idea that maybe these are two different forces, that the looping versus compartmentalization is two kinds of different forces, that one keeps the other one in control. What was really cool for me reading when this paper came by Xiaohui Zhang's group at Harvard was that all of these data I've been talking about is, of course, high-C data and in bulk cells. And then they have done this beautiful chromatin tracing experiments, which then they can look at a single cell at a time. And then they have shown that even if you get rid of QASN, these blocks at these diagonal patterns or triangles when you rotate them, but they are squares when you don't, they are still there in each cell. But what's very interesting is that the places at which their boundaries are occurring, there's not much of a preference. When you take away chromatin, QASN, then the boundaries can open anywhere. And then when you combine 10,000 single cells and plot this, the boundaries can be anywhere. Whereas if you have QASN, then you have these regions that are going to be more likely to be boundaries, and then you see these in the bulk data. So this is kind of an introduction, and of course a lot has been happening in the past 5 years or so in this field, but just wanted to let you guys know about what looping is about and what people are thinking right now. So we have even before some of these papers, we have when the first IC datasets started coming, we started thinking about, okay, how can we best analyze these datasets? Because with every genomics data, there is confounding factors and biases and other things that you need to worry about. And the initial methods generally, do not go into there, and then later these things are developed. So we developed an algorithm back in 2014 with the very simple idea that essentially, if we looked at the genome and where there's sequencing depth back then was the strongest, can we develop a way to look at the contact patterns and to identify these really specific and cell type specific interactions and loops between genes and other regions? So this is what we employed back then, and it has been actually highly utilized and cited because sometimes these simpler methods get better traction. In this case, it is because it's a method that is very flexible. So this background, we are learning from the data itself. We are not assuming a specific distribution. We are not doing anything fancy there. We are just saying the data, if you look at the data, it will tell you what the background should look like. And there are some statistical things that we are doing to make sure the null part of the data is separated from the real loops. But once you do that, then you can just simply use this, and then you can incorporate the biases and other things very easily into this formulation as well, which then for each pixel will assign a p-value statistical significance, which then you can multitest correct, because this is billions of tests literally, and then identify regions that are significantly coming together. This we did in human and mouse and malaria at the time, but it has been applied to essentially any genome that you can think of. Again, all because it's very flexible and it's an empirical null model. So more recently, though, with the increasing resolution of these high-C data sets and then the introduction of micro-C, which does improve the resolution also conceptually because it uses a different digestion system, we revisited this problem. So FitIC is a global background model. We look at globally what should be the background, and for each pixel we ask whether this is a bounded background. Whereas this one is a little bit different. So this came in when a PhD student at UC Riverside joined my group for a postdoc, and then he comes from a signal processing and image analysis background. So then when we look at these contact maps, I always think of them as a matrix. And then he started saying, okay, let's look at it as an image. And then there are a lot of tools in the image processing field that can be adapted for this type of problem. So one thing that he mentioned that is missing in the field is that think of these sunflowers, right? This is beautiful. And I've grown in such farms back in Turkey when I was growing up. So when you look at it, if you wanted to write an image processing algorithm that detects sunflowers accurately, what's going to need to happen is that it's going to have to deal with the scale issue, right? Something all the way in the back is a sunflower and something all the way in the front is a sunflower, but they're of very different scales. So the theory in computer vision that deals with this is called scale space theory. So essentially, scale space theory is, I will show you in the contact maps, but it does take an original map, original image, and then it does smooth it with a different Gaussian parameter. So it creates you a set of images that are smoothed to different extents. So then what it does is that you can take a difference between each of these different smoothed versions and get this difference of Gaussians. These are very easy to, manipulate. I mean, the Gaussian assumption and then a bunch of other statistical properties make sure that we are not introducing some random dots and points that are not there in the image. So it does limit the false positives. But when you get this difference of Gaussians, then you can do a computation of local maximum both within a given scale and across the adjacent scales. And then what this tells you is that not only that there is likely a loop here, but also that loop is most pronounced in the specific scale. So it allows us really to not only detect the loops, but to detect the scale of the loops, where if a loop is narrow and involving two pointy regions like this, or more broader between the two regions, we can capture them. So this has been extremely useful and extremely powerful because compared to the state of the art, the algorithm that everyone uses, which is a little bit data hungry, it doesn't work with lowly sequenced genomes. We were able to essentially find pretty much 80%, 90% of what this algorithm is able to detect in terms of loops, but much more, right? When you do this, of course, then you really need to show the ones that you are now finding are de novo, and we are doing that. I have some data for that. But another thing that was nice is that not only that, but we also compared to this algorithm, we also improved the reproducibility between replicates to 80%, where this was more like 60 to 70%, which becomes really critical for downstream analyses. And when we looked at the one question that we asked, there is this high-C data that we have all analyzed and high-C data that we are familiar with. And there was this micro-C that was the new guy on the block, essentially. The fundamental difference between the two is that high-C you use restriction enzymes to cut the DNA, whereas in micro-C you use MNAs, which does cut whenever it finds nucleosome-free regions, if you think about it. So this does give you much higher resolution. Of course, you need to sequence this deeper too. But when we looked at the same cell type and the high-C data versus micro-C data, what we have observed is that micro-C really does capture 90% of what high-C captures, but also much more. So this has been then validated in the field, and recent work points to this as well, that a lot of these loops that are micro-C captured they are between regulatory elements, and they are very important for gene regulation. So when we look at this is an aggregate plot of these peaks. So think about 36,000 such small matrices centered on each loop that we call, and then we take their average. As you can see, micro-C is like the signal to noise ratio is much better. It enriches that. So you have these more point loops here. And then when we looked at the genomes, when we forced high-C and micro-C to call the same number of loops, you can see that the high-C will go towards these bigger and broader loops, whereas micro-C is going to go for these smaller and darker loops. And then when you let the micro-C identify however many loops you can identify, as you can see, it captures everything that high-C does and more. That's very interesting and important for us. So one thing that allowed, and this is a very efficient algorithm, Abbas was able to implement it in a way that this runs in a couple minutes per chromosome for these extremely large data sets. So, and by extremely large, I mean like this is 1 kilobase. So if you think about the human genome as 3 billion base pairs, then you might be talking about a 3 million by 3 million matrix, and of course the chromosomes and so on and so forth. But it is a very big matrix that you are dealing with, extremely sparse. So what happened was that this enabled us working with resolutions that other tools haven't been able to scale, such as 1 kb resolution, and that enabled us to zoom into places that other tools and other papers have completely ignored, such as within 20 kb, because there's so much noise there due to physical proximity or linear proximity of the DNA that other people have essentially, including us in the past, we have refrained from really going and looking into that, thinking that we might not be able to identify specific groups there. So more recent paper from a group at Berkeley, Robert Tijan, so they have utilized our tool with their micro-C data and have studied the role of these specific proteins, that some of which I mentioned are important in the creating the DNA loops and domains. So what they have done is they have found 75,000 dots and loops, and they have done it in a way that they were able to, using this extremely highly sequenced micro-C data, they were able to go down to a couple hundred base pair resolution. And as you can see, our algorithm was able to really find these patterns of, enrichments that are corresponding to loops. So another, again, like the field is going towards more and more high resolution assays, generally genome-wide, but this was from Anders Hansen's lab at MIT. There again, they have come up with this region capture version of the micro-C. So instead of sequencing the whole genome, they are capturing a region of their interest, and they sequenced it in a way that, for these regions, in a way that if you, had a genome-wide, it would be 200 billion reads, right? So for, I don't know if I should put a price on this, but this would be, let's say, almost $1 million if you did a genome-wide for one cell type, one condition. So then what you see here is that, again, Mustache was able to identify these loops, these patterns that are only appearing at this ultra high resolution. So we believe that we kind of have a good way into this new set of experiments that are coming up that are pushing the envelope in terms of resolution at which we can find loops. And we think our tool is essentially going to be very critical in that. So this was work by Abbas, and then Tuvan was a student who came from Sabancı one summer, and then he really extremely helped us as well, publishing this paper. And Stefano Lonardi was also involved towards Abbas's PhD advisor. So then we went on to, then the data sets or the assays have evolved, and then so did our tools. So we have developed a tool called FitHighChip for analyzing some data that comes from an assay that's called HighChip. HighChip is essentially like Hi-C again, so you're trying to understand the 3D genome organization, but you want your sequencing money to be spent on regions that you care the most about and not on other places. So essentially then you have an antibody pull down step where whether you're interested in active histone modifications or a specific transcription factor, places it binds will be highly overrepresented. So this was discovered back in the 2016s, and then unlike the high C maps that I have shown you, this one has this pattern that it kind of has these enriched regions and then the background regions. So the same methods that we developed for high C had the assumption that the background is going to be more or less uniform and was utilizing that. So we had to develop another version of it, which essentially can take such contact maps and then turn them into these loops here. Here you are seeing a promoter and enhancers that were validated to impact the expression of this promoter. So if you are given this kind of a contact map, what you want to really come up with is these loops that are linking the promoter to these functional enhancers and nothing else and nothing less and nothing more. So a very talented postdoc, Surya Bhattacharya, he has developed this method here, which I'm not going to go into details, but kind of relies on the same idea, but then has a different way of dealing with the biases through a regression method. And then also deals with these indirect loops and kind of like finds where is the likely, anchor of these loops rather than reporting all of these. So this has allowed us to hit a balance between specificity and sensitivity of loop detection, because again, state-of-the-art previous methods, if you have given this region that I mentioned to you, that there are multiple validated enhancers, one of these would have found only the strongest one here. Another one that was in nature methods back then, it would essentially link everything to everything, whereas we think we have hit the right balance. And this is just one example, and we have, of course, the whole quantification of this in the paper, that we believe that this is a good way to identify such loops. So this is, of course, online, and Surya and with some collaborators at LGI, we have applied this to immune cells, and then this was in the context of just characterizing different immune cells and the disease-associated variants, and then later on, the same resource has been very helpful for us to look at when the COVID data came, COVID GWAS data came, identifying risk variants for severe COVID. We were able to then use these looping maps and identify the gene targets of such genetic variants, which gave us some ideas as to what are the actual genes rather than the regions that are important in immune cells for differences in COVID severity. So that's I guess, like, we can call it part one of my presentation. Now moving on to the part two, we have done, again, as I mentioned, went back to this chromatin compartments. And then this happened again with a talented postdoc and a high school student back then when he joined, that they started this project. How can we compare compartment calls across different conditions now that we are having more and more high C data sets every day, and it goes beyond this pairwise comparisons? So again, these compartments are those two big chunks of regions, these flat pattern regions that like interacting with each other but not across. And then generally, as conventionally, we call A compartment the open chromatin regions and B compartment the closed chromatin regions. And they fall out from this matrix. You take this matrix, you normalize, you correlation transform it, and then you do essentially principal component analysis or eigenvector decomposition on this. And then the first eigenvector does give you the positive and negative signs, these A and B compartments. So their identification hasn't been much of an issue, but when you look at it, then across different conditions, those values may not be scaled. The sign is completely arbitrary. Sometimes you put it in one method, and this is plus and this is minus, and another package, and it's completely arbitrary, which makes sense. Then you need to really flip it. distinguishing between technical and biological variations. So essentially using the replicate information hasn't been properly done, and then the comparisons were mainly limited to pairwise, and then now we have many data sets and time series and so on and so forth. So with that motivation, we have developed a method that's called DC-IC, differential compartment analysis of IC data. So it starts from these contact matrices or these compartment calls, and then after some normalizations, it uses this distance called Mahalanobis distance, right? So this is a generalization of a 2D Euclidean distance to multidimensional space. And then by using this distance, we are able to both look at within replicate and across condition distances and come up with statistical significance, again, of these changes in the compartments across different conditions. So we have of course shown it in pairwise comparisons that this is finding you the right kind of changes and consistent with what's out there. But then we applied it to a three-way comparison. Then we went to 13 different cell types in mouse, blood cell development, white blood cell development, if you think about it that way, or immune cell development, and then also a recent IC data, single cell IC data that came from studying developing mouse brain. So what we have learned is that this is loaded, and I'm not going to, of course, go through this, but hematopoietic stem cells going all the way to different lineages, common lymphoid progenitors, granulocytes, megakaryocytes. So you see a lot of changes in the compartments, and then what our method actually is able to do, I'll first show you this, that it's of course able to find cell type specific compartmental changes, such as this one where this region and this gene is active in the granulocytes. And then as you can see, when you look at the compartments, it's only here, and then you can see that we assign it a statistically significant, very high or low, I guess, p-value here that determines it's significantly different. This is another gene where the traditional methods would have failed to find because it's in the A compartment across all of these different conditions. It's an important transcription factor, and it's expressed throughout hematopoiesis, but to different extents in different progenitors and different cell types. So it's kind of like highly fine-tuned. And as you can see, our method captures this as a statistical difference between these conditions, highlighting that not only things that flip like this one and obvious, but fluctuations is also important, which you can capture from this multi-dimensional data analysis. So maybe I will not go into this too much, but what you are seeing here is again doing it in the mouse brain development for cortex and hippocampus, and 6 time points each. When we looked at it as early, middle, late in a given tissue, we found these compartmentalization patterns and clusters of them that change concurrently. And again, they correspond to regions such as this one, where the compartment goes from active to inactive, or also this one, where the compartment goes in accordance with the role of this gene and this region in the early and late development. Compartment gets more and more active, or different changes significantly. That highlights an important feature of this gene. So again, this was developed by Abhijit, who's still in the lab, and Jeffrey, now undergrad at Harvard. And then they have developed not only a really nice package, but also super easy to browse IGV browser visualizations that immediately fall out of the application of the method. And this was published last year. So going back, I kind of I could have talked about some other published work and all of that, but I really want to talk to you about something that is new and that I'm excited and that I think is holding a huge potential in terms of what we can do with all of these computational tools in a context of disease. In this case, it's pediatric cancer. How we can use it to find interesting and novel targets and potentially inform new therapies. So As I mentioned, we can go back to these Hi-C maps or contact maps, and then go after patterns that are unexpected or abrupt, or that are not explained by the regular 3D folding. So when you look at this, you may realize that some regions are more faint and some regions are more stressed. And then when you look at the coverage, you will see a difference there. Overall coverage, essentially, marginalized counts per row or column here. And then on this side, this is a cancer cell line, breast cancer cell line. And then on this side, what you can see is translocate, well, I shouldn't give it away, but interchromosomal patterns that are not supposed to be there, like between two chromosomes, strong interactions that are as strong as within chromosomes that are not supposed to be there. So back when I first joined, this was our first paper with Abhijit, where we have said, okay, let's utilize these patterns and then identify genomic rearrangements. And then we utilize these patterns to identify copy number variations. And in this case, this is a translocation where arms of two chromosomes are exchanged, essentially. And then that leads to these patterns. So we developed algorithms back then for this. But while we were doing that, we were thinking, okay, this is like, these may be easy cases. And we know that there are more like complex rearrangements. What can we do? Or what would be a good model to study whether our methods are useful for solving something that's more complex? And then with some collaborators, we ran into this data where there is a mouse model, mouse embryonic stem cells, that to study Down syndrome, which is a trisomy of chromosome 21, scientists have tried to insert human chromosome 21 into mouse cells. And of course, the chromosome is big and you cannot stick it into there immediately. So how they achieved that was irradiate human chromosome 21 and then transfer those irradiated pieces, which goes into mouse cells. And then when it goes into mouse cells, somehow it finds its pieces back together. But you can think of it like it's finding its pieces back together is done in a hurry, that it's kind of like putting everything in a bag. And it's not putting them in the right order. So they are coming into an order that is completely rearranged and reshuffled. So when you then have Hi-C data from these mouse cells, when you look at the human chromosome, which you can distinguish because the reads will map to human genome and not to mouse genome, when you look at the Hi-C data for this chromosome, you can see all of these patterns. That wouldn't be there if that was a normal chromosome. On the left, you're seeing a normal chromosome and how Hi-C map should look like. So then this was our challenge. We are like, okay, let's see whether we can go into this and then develop a computational method that first detects all of these breakpoints and all of these small fragments, and then essentially, just like Rubik's Cube, shuffles them back into place, into the right order, and then gives us, in the newly created genome sequence, gives us something that, is the right sequence. So Abhijit has developed an algorithm there, and this was not a very difficult one. We used genetic algorithms here to find these correct rearrangements. And then we started thinking, okay, this is very important, the complex rearrangements, and we can solve them. But this is a mouse model where, solving this may or may not be as valuable. Can we look at something where these kind of rearrangements happen in reality and in relevant human diseases? So that rearrangement that I just mentioned, when it happens on its own, it's called chromathripsis. Essentially, it's a catastrophic event that hits a chromosome. And then a chromosome like this one, when this happens, which we believe happens immediately, not like one by one, when this happens, it does get shattered into pieces, sometimes tens or hundreds of fragments. And then, as I mentioned, then some of those pieces get lost, some of those pieces get amplified, and some get put back together, but not in the correct order. And then this is progression towards cancer after that. So where does this happen? So the interesting thing is that when we first started studying this, the common knowledge was that, well, yes, chromatopsis is an important event, but it's only happening in like 2 to 3% of cancer genomes. So it's kind of like a rare event. And then the Pan-Cancer Atlas came with a paper that said, no, it was just the way that we were looking at these things with the old technologies. Now that we can look at it with whole genome sequencing and newer technologies, actually chromatopsis happens in 30 to 40% of cancers. So all of a sudden, the profile of what we are studying has gone up by tenfold, and we already, by then, we have identified the one kind of disease that we wanted to study, which is the pediatric B-cell leukemia, and a specific high-risk subset of it, which there's chromatopsis. So that subset is called IM21. And then if you look at IM21, it is the one with the worst survival and the highest relapse risk. Luckily, this is from quite some time ago. After realizing this, people and doctors have started treating these kids more aggressively, and an aggressive treatment actually makes a survival chance much, much higher, although it is still one of the worst ones, and relapse rate still remains high. It's a very rare category within all B-ALLs, pediatric B-ALLs. Only 2% of the kids will have this, and those are the ones who are not going to fare very well comparatively. And it's marked by that chromatopsis that's again on the chromosome 21. So what did we do? We collaborated with clinicians and other scientists, and we have gathered the pilot set of samples. These are difficult to get, like one hospital or one center is not going to be able to find you. Even for 5 of these cases, you really need to look for bigger collections of them. So we were able to get a pilot 9 patient samples, and then 10. Again, these are control, but these are leukemia, but non-IM, not the one that we study. So we have done RNA-seq, whole genome sequencing, the Hi-C, the experiments that I talked about, the single-cell RNA-seq, and more recently long reads with the PacBio platform on all of these samples. So what we have first confirmed is that really these kids with the IM21, their chromosomes are rearranged, and there are large amplifications that are happening in the chromosome 21. And a few of the control cases actually seems to have that. When you go into the high CMAPs we have generated, you see this kind of pattern. When you zoom in a little bit, it's clear that you have these shufflings and you have these small fragments, and they're all over the place. Something for us to resolve. When we looked at our small set of samples with a huge cohort study of 700 pediatric samples, we have seen that our cases, most of them are co-located with these high hyperdiploid cases and pH-like subsets that were identified before, but a couple of them corresponded to these ETV6, a specific gene, and mutations of it. And then when we look at it, we see deletions for those 2 samples. We see deletions of ETV6. And one of our samples corresponded to PAX5 alterations group. And then when we look at this sample, we saw that it does have a PAX5 deletion also in our case. So we are trying to just put our findings in the context of what's been known and then also what's novel. So large amplifications in chromosome X is what we found, and then chromosome X deletions have been found before, but I think this is the first time we are showing that there are large amplifications of this chromosome. And more interestingly, we have found a region where we see this same pattern, same rearrangement pattern across 2 different samples. And this is recurrent, and in this case, it doesn't come with any other interchromosomal translocational rearrangement, whereas in this sample, it's very interesting that it does have a rearrangement also interchromosomally. And in another patient sample, there is no rearrangement in this region. So this may have important implications in terms of stratifying patients into disease groups or their therapies. And then another rearrangement we found is a translocation. That's unbalanced translocation. So essentially, a piece of chromosome X gets amplified and sticks into the end of the chromosome 12 and creates this pattern. So it's amplified. It's not deleted in its native location, but it's there. But it's also goes and translocates. It's this little piece on chromosome 12. So when we look at these rearrangements in a single-cell RNA-seq data and whether they are clonal or subclonal, what we have identified was that some of them are subclonal. So there are two different kinds. You'll get the same patient samples, and then none of them are happening in control, fetal bone marrow B cells here. When you look at these when you look at this one patient and all of its single cell data, you can see that the chromosome X amplification happens only in a subset of cells and not in the others. So this does tell you that you really need to look at, in cancer in general, you really need to look at the clones and subclones and then try to understand what comes first and what comes later. So then we looked at the single cell data of these different patients, and as you can see, IM patients group in one place and all the other control patients and control cells grouped separately. We have then went on to looking at the gene expression, and then we have identified differential genes that demarcate this IM21 and separate it from the other cases of leukemia. And obviously, chromosome 21 had certain genes that where this is happening, but we have found, very interestingly, genes that are not on chromosome 21, but are also markers of this specific disease subset. So some of these targets are very interesting, and we are now working on them. Actually, two of them, we have some follow-up experiments and follow-up work. So I'm very hopeful that we will be able to come up with new leads for kids that are of this subset, and then maybe potentially targets that are going to be useful for them in the therapy. So this project is essentially all spearheaded by Abhijit in the lab. So when we needed, Adam joined. We had a small wet lab, and we still have it. Adam has generated some of these data. Vijay's lab has generated a lot of the single cell data, and also RNA-seq and whole genome sequencing. Now that we have a lot of data, Abhijit needed help, so Laura, undergrad student from Mexico, she joined the lab and now helping Abhijit. And we were able to get some funding from foundations, a Spark grant and a Conrad Prebys Foundation, and the samples we gathered from children's oncology groups. So we are thankful to all of them. Yeah, but with that, I'm going to end, and hopefully we will have time for questions. I really thank all my lab members and my collaborators. We are a very diverse lab. As you can see, we have people from all over the world. I really value this and appreciate that I'm able to talk to people with such different perspectives, and our funding, and of course our GitHub, where there's a whole bunch of very useful tools if you're in this field. And also thankful to our collaborators who helped us apply all of the methods that I'm talking to you about today into different kinds of domains and made the difference in such domains. So with that, I'm hoping everyone is still here and not asleep, and we have some questions.
Thank you so much, Dr. Ferhat Ay. We appreciate for your very informative presentation. I'm moving on to the question and answer section.
All right.
I'm continuing with the first question. Can 3D modeling studies use the data to characterize promoter-enhancer interactions in single cells or to validate and interrogate predictions about genome folding?
That's really nice. So yes, the short answer is yes. The longer answer is that the single cell data doesn't have the resolution to really go after such fine scale enhancer promoter-like interactions at this moment. But as I have shown, imaging has gone a long way in imaging high throughput DNA. So it is now showing us certain enhancer promoter interactions at the single cell level, and people have started like going from single cell data to such loops, but it is very difficult and challenging at this point.
Thank you so much. I'm moving on to the next question, coming from the YouTube participants. Profiles of chromatin interactions captured by the Hi-C are formed by the joint action of different mechanisms. So does this profile to result in extreme complexity of Hi-C libraries?
Yeah, good question. It does, yes, exactly. So, and then that extreme complexity is why we need more sequencing than, you know, a couple, maybe at least an order of magnitude more sequencing than other assays such as RNA-seq, ChIP-seq, etc., which you can do with tens of millions of reads. Whereas here, the complexity of different types of loops is said mediated by different proteins and different mechanisms, and also the number of such things. And of course, also the dimension, right? I mean, when you do ChIP-seq, you have an array that you are trying to fill with numbers. When you do Hi-C, you have like a 2D n by n rather that you're trying to fill with numbers. So extremely complex libraries, and then even more complex with the micro-C and other new techniques, and they need to be sequenced, and their analysis also becomes a little tricky if you don't know what you're doing. So that's where we are hoping that we can help.
Thank you so much. The next question is it possible to predict chromatin interactions for different cell types lacking experimental Hi-C data?
Oh, great question. That's a bioinformatics student, I would think, who's asking this question. So yes, to a certain extent, it is possible. And there have been papers that are using tensor decomposition and other deep learning models where they use correlations of other data sets such as ChIP-seq and so on and so forth, or chromatin accessibility, to determine cell type specific Hi-C interactions when you don't have the Hi-C data, or when you have it and you can cross compare. So it is to a certain extent, but my approach to a lot of these can we predict X from Y kind of problems is that it does teach you a lot, and they're good projects for students to learn how things work. But it is very difficult to convince, let's say, a biologist or immunologist to say, oh, like you don't have Hi-C data in that. Let me go through this whole process of doing X, Y, Z and all these super complicated methods and predict it. So they would rather generate it, right? It's kind of, for me, it is difficult in that sense to justify just using these predicted, loops or predicted contact maps when it is possible to actually generate the data. But there are cases when this would be very useful, when there are not enough cells, or you're not able to access them for this experiment, whereas you can perform the others and then use the other experiments to infer that. There are such cases, but they are not as common. Great question, though.
Thank you for your answer. I'm continuing with the other question. Too many methods source could be the reason of chaotic information source. How can we be sure about reliability of genome data?
I have no idea what this question is trying to ask, but reliability of genome data. Can I skip this one? Because I mean, maybe it's the wording.
Of course. I'm continuing with another question. If we fully reached out the genome mapping for gene modeling compounds within protein bindings and epigenetic factors, it can be scaled up chromosomes modeling to biologic regulation viewing.
Yes, I guess if you reached out genome mapping for gene modeling, so we are doing genome mapping and gene modeling and all the protein binding epigenetic factors. So if the question is whether we can use those to do the 3D modeling of the chromosomes, then the answer is pretty much the same as what I mentioned, that yes, you can, and yes, there are algorithms, again, in tensor decomposition, etc., using that one-dimensional signals, epigenetic factors, etc., to predict, to a certain extent, the cell-specific regulation or regulation weaving, I guess. So, yeah.
Thank you so much. And moving on to the next question. It is crucial to improve the computational performance of 3D modeling methods in understanding the genome structure-function relationship. Could we achieve this goal by developing highly parallel code that scales well across several CPUs or GPUs, or by creating our lattice models that allow for much faster simulations and parameter scanning?
Yeah, so yes, the answer is yes to this. And again, we want to, first of all, it's like software engineer, hardware engineer kind of thing, right? So we want to, first of all, come up with the right kind of algorithms and methods that they are in their own, like scalable and as efficient as possible. But when that fails for maybe like structural modeling and so on and so forth, then we really need to go into these. And then some of the algorithms we have developed are efficient enough that and that they are these shamefully parallelizable, that you can run it across different chromosomes separately, and then that's your parallelization. But some more complicated models and maybe molecular dynamics simulations and so on and so forth, they need these type of things. And more recently, I haven't talked about it, but we have some data, some method development in single cell RNA-seq analyses where we really needed to go into GPUs and parallelization and learning parameters and tracing the parameter space much faster with the GPUs. So all over the place, this is useful.
Thank you so much. How can we get in depth of sequencing to enable robust and sensitive detection of individual chromosomal contacts?
By spending more money. So you just need to sequence more, and it does cost a lot of money. So there is a recent paper that's suggesting for proper differential analysis of contacts that you need to have at least a couple replicates, of course, but also like 6 billion reads per condition. 6 billion reads is like almost the whole NovaSeq S4, and that is like 30,000 plus. So it's just money, essentially, more sequence.
Thank you so much. We will appreciate for your answer. Another question from you, the participants, is Ferhat Ay C2 make any assumptions about the size or the structure of the genome?
It doesn't actually, yeah. That's a good question. I mean, neither Ferhat Ay C nor Ferhat Ay C2, it doesn't make any assumption. It can be any genome, any, as I said, people have used it for yeast, for cotton, for, I don't know, mouse, human, and all of that. It does infer the background itself from whatever data you give, and that's one nice feature.
Thank you so much. I'm continuing with another question. Which methods the best for genome-wide and promoter-wide detection of potential gene regulatory interactions at high resolution using low cell numbers?
Yeah, that's a good question. So there are the major methods, especially when you think about promoter or like regulatory element centric, there have been other than HiC, of course, there have been HiChip, and also promoter capture or region capture or enhancer capture. You can talk about capture-based chromosome conformation assays, and all of them have improved to a very large extent in the number of cells that they need. So the capture experiment started from 100 million cells. Now I think they are down to about 1 million. HiChip has started from a couple million. It's down to a couple 100,000. So my personal favorite is HiChip because we have used a lot of HiChip data, and we believe that the dynamic range of chromatin contacts that you can capture from HiChip using low cell numbers is really sufficient to detect interesting biological differences. But yeah, depending on your applications, it may change.
Thank you so much. We would appreciate. Another question is Ferhat Ay be able to call loops between peak and non-peak regions?
Yes, that's that we allow, because there are very interesting and important interactions we think are happening, and they have different chromatin marks on the two sides. And one anchor may have an activating mark, but the other may be missing it, but it may still be important. So if we only require that the peak-to-peak interactions are discovered, then we would be missing such, and then that's why we develop. But for the people who are really interested in maybe like these structural loops between two CTCF sites, etc., we have peak-to-peak version too, so it's just a user choice.
Thank you so much for your response. Can DC-Hi-C also be applied to the comparative analysis of other coarse-grained genome-wide signals, such as replication timing and lamine association?
Yeah, someone read the paper, it sounds like. Okay. Yes, so we mentioned this in the paper, and that signals from anything else that covers the whole genome, such as the Hi-C compartments and replication timing, lamine association, and other assays that you can think of. Yeah, it's essentially a multi-way comparison tool for genomic signals, and it does, and we don't have it in the paper, but we have applied to replication timing data and got pretty good results in working on that.
Thank you so much. I'm continuing with another question. In Hi-C and other methods, is the presence of each loop associated with higher gene expression? How can we differentiate this? Yeah,
nice question. Something that the field is also always thinking and asking about. The answer is that not always, right? Generally, when you do an aggregate analysis, generally the presence of a loop, especially if it's from an activating chromatin kind of H3K27 acetylation, etc., kind of histone modifications and their high chip data, then you do have this correlation of increasing interactions correlating with increasing gene expression. But again, there are other loops, there are silencing loops, there are repressors in the genome, there are insulating regions, and looping to them may actually decrease chances of gene expression. So how you differentiate this is through integrating it with the gene expression data and the looping data. But how you can model this is very difficult. Like a genome-wide model, like certain assumptions will not work for every single loci. So.
Thank you so much. As a last question, is the any methods for distance-dependent analysis of Hi-C interactions within and between chromatin compartments?
I see. Yeah, I'm not sure exactly. This person can reach out later, but so some of the methods that we are talking about, regardless of the compartments, they do allow for the distance dependence and its correction, but nothing to the effect, if it's what I'm understanding, nothing to the effect that the distance between compartments and then those maybe aggregate plots and other things that you are doing with compartment-to-compartment interactions being accounted for their distance. Not that I know of. Of course, we do regular normalizations and all of that, but nothing that's properly done.
Thank you so much. And I would like to ask something about what advice would you give to students studying in scientific fields?
Yeah, I mean, in scientific fields is broad. I think I can talk about maybe bioinformatics and students who are in the same kind of track and mindset that I have been in. And the one thing that I'm learning as we progress through our careers is that bioinformatics is a very exciting discipline, but it is not a discipline where you can stick to one hammer that you have and then expect nails coming and then you hitting them, right? So it's not a place where you can just rest easy because you have developed something and then it will work forever. So it is extremely dynamic, and the right kind of bioinformatics student will have the mindset that they will come and they will work on the interesting problems of the day and maybe develop some very important tools. But tomorrow or next year, they might be working on something quite different, and they should be excited by it rather than being afraid of it. So if you have that kind of mindset, I think then you might be the right type of person in bioinformatics, that you should be open for change and dynamism and collaborations and being scooped and your tools being kind of obsolete within a couple of years and so on and so forth. If those are okay with you, then bioinformatics is the right place. But it's not a place where you can come and rest easy, and it works at a very fast pace compared to wet lab and other things. So you may have an idea, but there's a lot of other smart people. The moment some data comes, you may have a good idea, but a lot of other people will have that good idea too, right? There's lots of smart people. So you really need to act fast and then execute. So you really need to have those skills, in our field at least.
Thank you so much. On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation. Thank you
Thanks
so much.
Thanks a lot, Nazlı. And then thank you to the whole group. And this has been I wasn't able to watch it with the time difference and all of that, but I scanned through the YouTube, and it seems like an amazing lineup, such a diverse set of people. And I'm glad that you're kind of like connecting all our scientific researchers working in other countries back to our country, and then helping our students, motivating them. Hopefully, if they didn't understand anything from this, the one thing that they can take away is that if they're motivated, I'm sure they are smart. If they're motivated and if they're up for the challenge, there's no limit. So good luck to everyone.
I wish you healthy days.
Yes, you guys too. Take care.